You know how we're all familiar with the concept of extinction? When a species disappears, as in goes the way of the dinosaurs, it's gone forever. Or at least, that's what we thought. But what if I told you a team of scientists, artists, and perfumers have found a way to resurrect a piece of these lost species, their scent? We're talking about a project aptly named Resurrecting the Sublime, a collaboration between Boston-based biotech company Ginkgo Bioworks, artist Alexandra Daisy Ginsberg, and smell researcher Cecil Tolas. Together, they've achieved what one researcher called Jurassic Park, but for perfume, extracting DNA from preserved, extinct flower specimens, analyzing the genes responsible for scent production, and recreating fragrances that haven't been smelled by human noses in over a century. Why does this matter? Well, beyond being a cool party trick, this project represents a fascinating intersection of advanced biotechnology, artistic expression, and environmental awareness. It's giving us a sensory connection to what we've lost through human activity, while simultaneously showcasing the incredible possibilities of modern manufacturing and synthetic biology. Welcome to Apprentice.io's Industry Insights. I'm Frank Maggiore, and today we're diving into a story that's equal parts science fiction, cutting-edge biotechnology, and artistic expression. Imagine walking into a museum exhibit and smelling a flower that hasn't existed on Earth for over a century. Sounds impossible, right? Well, hold on to your lab coats, because that's exactly what's happening right now. And let me tell you, the technological achievements here are mind-blowing. We're talking about taking degraded plant specimens collected over 100 years ago, extracting fragments of DNA, piecing them back together, and then engineering yeast to produce scent molecules that no longer exist in nature. If that's not a manufacturing revolution, I don't know what is. Today, we'll explore how this fascinating project works from start to finish, from finding preserved specimens in dusty herbarium folders to the cutting-edge genomics and biomanufacturing that bring these scents back to life. We'll look at the crucial role of manufacturing execution systems in making this precision bioengineering possible, and explore what all this means for the future of both perfumery and biomanufacturing as a whole. Ready to take a journey through time with your nose as your guide? Let's dive in. Let's start with the stars of our show, 3 remarkable flowers that disappeared from Earth due to human activity, but have now had their scents brought back from the dead. First up is the Hibiscadelphus wilderianus, also known as the Maui hakuaewe. This flowering tree native to Hawaii was last seen alive around 1912. That's the same year the Titanic sank. It grew up on the southern slopes of Mount Haleakala on Maui in ancient lava fields that were decimated by colonial cattle ranching. The tree was so rare that only a single specimen was ever documented. A botanist named Garrett Wilder collected samples from the last known tree, and it's those very samples that made this resurrection possible. Then we have the Orbexilum stipulatum, or Falls of the Ohio scurfpea. Scurfpea sounds like it would be a venereal disease for pirates, but it's just in the family of legumes. This was a small flowering plant from Kentucky that hasn't been seen since 1881. We're talking about a plant that disappeared while Billy the Kid was still roaming the Wild West. Its native habitat was a single location, Rock Island on the Ohio River. When humans built a dam that flooded the island in the 1920s, they erased not just the plant's home, but every trace of its existence. Our third floral ghost is Leucadendron grandiflorum, commonly known as the Wynberg conebrush. Native to the area around Table Mountain in Cape Town, South Africa, this plant fell victim to colonial vineyard expansion. It was last seen in a collector's garden in London in 1806, which was during Thomas Jefferson's presidency. An interesting twist with this one, researchers discovered there may have been some confusion in the historical records with 2 different specimens labeled as the Wynberg conebrush, throwing the exact identification into question. What do all these plants have in common? They were all driven to extinction by human activities, cattle ranching, dam building, vineyard expansion, and all that remained of them were pressed, dried specimens carefully preserved in folders at the University Herbaria. That's where Christina Agapakis, creative director at Ginkgo Bioworks, enters our story. In 2016, she visited Harvard's vast collection of preserved plant specimens with a wild idea. Could they extract DNA from these samples and reconstruct their scents? The Harvard Herbaria is like a plant library with over 5 million specimens, some collected hundreds of years ago. Agapakis and her colleague, Dawn Thompson, went through the archives searching for extinct plants. They found 20 extinct species in the collection and were able to take samples from 14 of them. Ultimately, they focused on the 3 I just described. These pressed plants and manila folders were all that remained of species wiped out by human activity. But as it turns out, even in death, these plants still had a story to tell and a scent to share. They just needed a bit of high-tech help to tell it. Now let's get into the nitty-gritty of how you actually resurrect the scent of an extinct flower. And trust me, this isn't as simple as finding a recipe in an old cookbook. It's more like trying to reconstruct a symphony when all you have is a few scattered pages of the score. The process starts with something that sounds straight out of a sci-fi movie, extracting DNA from plant specimens that have been dead for over a century. The ginkgo team took tiny tissue samples from the dried plants at Harvard's Herbaria. We're talking about plants that were collected in the early 1800s and 1900s. These samples were old, degraded, and extremely precious. But here's the problem: when DNA sits around for 100 years, it doesn't exactly stay in mint condition. It breaks down into fragments, making it incredibly difficult to piece back together. This challenge was so great that ginkgo had to partner with paleogenomics experts at the University of California, Santa Cruz. These are the same types of specialists who work on extracting DNA from ancient fossils. The UC Santa Cruz team crushed the plant samples into powder and used special chemical techniques to extract whatever DNA fragments were still viable. Think of it like trying to recover data from a corrupted hard drive. Painstakingly, detail-oriented work. Once they had these DNA fragments, the next challenge was identifying which genes might be responsible for producing scent molecules. Plants create their fragrance through enzymes coded in their DNA. These enzymes help produce terpenes, esters, alcohols, and aldehydes, the chemical compounds that make a rose smell like a rose and a lily smell like a lily. To fill in the missing genetic information, the ginkgo team compared the fragmented DNA with known sequences from related living plants. They were looking specifically for genes that encoded enzymes involved in creating fragrance molecules. It's like having a partially completed crossword puzzle and using the surrounding words to figure out what's missing. Once they identified the potential scent-producing genes, they needed to test if it actually worked. This is where real bioengineering magic happens. The team synthesized these gene sequences and inserted them into yeast cells. Wait, yeast? Yes, the same stuff you use to make bread and brew beer. Yeast is the workhorse of the biotech world. It's easy to grow, reproduces quickly, and can be engineered to produce all sorts of compounds. When the engineered yeast ferments sugars, it doesn't just produce alcohol like it normally would. It produces the fragrance molecules encoded by the extinct flower genes. It's essentially a microscopic factory producing molecules that haven't existed in nature for over a century. To identify what compounds the yeast were producing, the team used a technique called mass spectrometry, which can identify molecules based on their mass and chemical structure. This gave them a list of the potential scent molecules that the extinct plants might have produced. But here's the fascinating twist. Even with all this advanced technology, they couldn't know for certain exactly what proportions of these scent molecules were in the original flowers. It's like having all the ingredients for a cake, but without the exact measurements. This is where the art meets the science. Smell researcher Cecil Tolas took the shopping list of molecules and created different fragrance combinations, essentially making educated guesses about how the original flowers might have smelled. This result isn't an exact replica of the extinct flower scents. It's more like a scent reconstruction, or what the team calls a smell hypothesis. It's our best scientific estimate of what these lost flowers might have smelled like, brought back through a combination of ancient DNA, cutting-edge genomics, bioengineering, and artistic interpretation. And that, folks, is how you revive a scent that's been extinct for over a century. Not exactly simple, is it? Now let's talk about what makes this whole process not just a cool science experiment, but a manufacturing revolution. Because what we're really discussing here is a whole new paradigm for how we make things. In this case, fragrance molecules that literally don't exist anywhere else on Earth. Traditional fragrance manufacturing typically involves one of two approaches: extracting compounds directly from plants, think rose oil from actual roses, or synthesizing them through chemical processes in a lab. But what Ginkgo Bioworks is doing here is fundamentally different. They're programming living organisms to be their manufacturing facilities. This approach is called fermentation-based manufacturing, and it's transforming how we produce everything from fragrances to pharmaceuticals. Instead of building giant chemical plants that require extreme heat, pressure, and often toxic solvents, Ginkgo is using microscopic yeast cells and just feeding them sugar. Let me walk you through how this manufacturing process works in practice. First, Ginkgo designs the production line by genetically engineering yeast cells with the resurrected genes from extinct flowers. This happens in their headquarters in Boston, which they call their foundry. Think of it as a biological factory where robots and automated systems help build and test thousands of different microorganism designs. Once they have the yeast strains that produce their desired scent molecules, the fermentation process begins. This is similar to brewing beer, but instead of producing alcohol, they're producing specific fragrance molecules. The yeast cells eat simple sugars and convert them into complex scent compounds. Ginkgo has developed sophisticated fermentation capabilities that can scale from small laboratory bioreactors all the way up to 50,000-liter industrial fermenters. For their work with fragrance partner Robertet, they've successfully scaled production to commercial levels. We're talking about producing enough material to actually use in commercial products. But here's where manufacturing execution systems, MES, become absolutely critical. When you're dealing with living organisms as your manufacturing platform, there are thousands of variables to monitor and control. Temperature, pH, oxygen levels, nutrient concentrations, and a whole lot more. A tiny variation in any of these parameters can completely change what molecules your microbes produce. For this type of complex biological manufacturing, advanced tracking and process management would be essential. A sophisticated production system, equipped with sensors to monitor bioprocesses in real time, could create what engineers called a digital thread, a complete, unalterable record of everything that happened during production, ensuring quality and consistency. The MES doesn't just collect data, it uses artificial intelligence to analyze patterns and optimize processes. If something starts drifting out of spec, the system can automatically adjust parameters or alert operators before the yeast cells even know that something's wrong. After fermentation is complete, the scent molecules need to be extracted and purified. This involves separating the molecules from the yeast cells and culture medium, typically through centrifugation, filtration, and various chromatography techniques. Again, the MES tracks every step of this downstream processing to ensure quality and consistency. What makes this approach so revolutionary is the precision and flexibility. Traditional chemical manufacturing often requires completely different equipment and processes for each new molecule. With Ginkgo's approach, the same fermentation equipment could produce wildly different compounds just by changing the yeast strain used. It's like having a printer that could print anything just by changing the digital file it's working from. This manufacturing approach is also substantially more sustainable than traditional methods. It uses renewable feedstocks, sugars instead of petroleum, operates at lower temperatures with less energy, and produces fewer toxic byproducts. Ginkgo's partner, Sumitomo Chemical, has specifically highlighted how this approach can help create products with a lower carbon footprint. The implications go far beyond just resurrecting extinct flower scents. This same technology platform can be used to produce flavors, pharmaceuticals, cosmetic ingredients, biomaterials, and a whole lot more. Ginkgo has over 40 products under contract with 20 customers, spanning industries from food to fragrance to pharmaceuticals. So when we talk about resurrecting the sublime, we're not just talking about a fascinating art project. We're talking about a showcase for an entirely new manufacturing paradigm that could help transform how we make everything from perfumes to life-saving medicines. So what happens when you've successfully resurrected the scent of an extinct flower? How does this fascinating science project translate into the real world? For resurrecting the sublime, the first application was artistic. The project resulted in a series of immersive installations that have been exhibited at prestigious venues around the world, including the Cooper Hewitt Smithsonian Design Museum in New York, the Centre for Pompidou in Paris, and the Cube Design Museum in the Netherlands. These installations are not just about smelling something interesting. They're designed to create a profound emotional experience that connects visitors with what we've lost. The scents are diffused in custom-designed spaces that include elements referring to each flower's lost habitat. Lava rocks for the Hawaiian hibiscus and limestone for the Kentucky scurfpea. As artist Alexandria Daisy Ginsberg puts it, the installation makes us contemplate a fundamental question. Since humans were responsible for these extinctions, do we deserve to smell these flowers again? It's a powerful reminder of our impact on the natural world and what's at stake as we face ongoing biodiversity loss. But beyond the art world, this technology is starting to make its way into commercial products. The fragrance industry has been quick to see the potential of these biotechnology approaches. Ginkgo has partnered with several major fragrance companies, including Robertet, a global leader in natural ingredients. In 2017, Ginkgo and Robertet announced the successful commercial-scale fermentation of fragrance ingredients, proving that this technology could move beyond the lab and actually produce marketable products. They focused on rose oil components and a family of lactone ingredients. Those are compounds that create creamy, fruity notes in perfumes. More recently, a Boston-based beauty brand called Future Society, formerly Archaea, launched a collection of 6 perfumes inspired by extinct flowers using Ginkgo's DNA sequencing data. Their bestseller is Solar Canopy, a vibrant sweet floral inspired by the extinct Hawaiian hibiscus we've been discussing, combined with contemporary notes like red currant and lychee. The commercial potential goes far beyond just recreating extinct scents. This same biotechnology platform can create novel fragrance molecules that don't exist in nature, or produce sustainable alternatives to ingredients that are currently harvested from endangered plants or manufactured through environmentally harmful processes. For example, traditional perfumery has relied on ingredients like ambergris from sperm whales, musk from deer, and certain orchid species that are becoming increasingly rare or ethically problematic to harvest. Bioengineered alternatives could provide sustainable substitutes. Fermentation-based manufacturing also offers advantages in consistency and supply chain reliability. Natural ingredients can vary significantly based on weather, growing conditions, and harvest timing. Lab-grown alternatives provide consistent quality year-round. The potential market impact is enormous. The global fragrance ingredients market is projected to reach $16.1 billion by 2027. As consumers increasingly demand sustainable and ethically sourced products, bioengineered ingredients are positioned to capture a growing share of this market. Beyond fragrances, similar approaches are being applied to food flavors, cosmetic ingredients, and even pharmaceuticals. Ginkgo's technology platform is industry agnostic. The same principles used to resurrect extinct flower scents can be applied to produce everything from vanilla flavoring to anti-cancer compounds. What started as a fascinating art-science collaboration is evolving into a commercial reality that could transform multiple industries. It's a perfect example of how cutting-edge biotechnology, when combined with creative vision and advanced manufacturing systems, can create entirely new market opportunities that weren't possible just a few years ago. So where does all this lead us? What's the future of bioengineered scents and the broader field of synthetic biology manufacturing? First, we're likely to see more extinct scents brought back to life. The Harvard Herbaria contains over 5 million plant specimens, including many extinct species. Ginkgo has already sampled 14 extinct plants and focused on 3 for this project, but there's enormous potential to expand this work. But the technology's applications go far beyond just recreating lost scents. We're entering an era where biotechnology could fundamentally transform the fragrance and flavor industry. Imagine custom-designed scents that have never existed in nature. Fragrances engineered molecule by molecule for specific properties. Want a perfume that lasts exactly 8 hours, then gracefully fades? Or one that changes its scent profile throughout the day? These could become a reality through synthetic biology. The sustainability implications are equally exciting. Many valuable fragrance ingredients currently come from rare plants or require environmentally damaging extraction processes. Bioengineered alternatives could reduce pressure on endangered species and fragile ecosystems. There's also potential for personalization at an unprecedented scale. Your personal biology affects how you perceive scents and how fragrances interact with your skin chemistry. Future biotech could enable perfumes tailored to your specific genetic makeup and preferences. From a manufacturing perspective, we're witnessing the birth of a new paradigm. Traditional chemical manufacturing often requires large, centralized facilities with massive capital investments. Fermentation-based approaches can be more distributed, with smaller facilities serving local markets, similar to how craft breweries operate compared to giant beer manufacturers. The role of manufacturing execution systems will become even more critical as biomanufacturing grows. When your factory consists of billions of living cells, each potentially behaving slightly differently, you need sophisticated real-time monitoring and control systems to ensure consistency and quality. We're also seeing the integration of artificial intelligence with synthetic biology. Ginkgo is already using machine learning to analyze vast data sets and predict which genetic modifications will produce desired results. As these AI tools become more sophisticated, the design, build, test cycle will accelerate dramatically. Regulatory frameworks will need to evolve alongside the technology. Currently, bioengineered fragrance ingredients face varying regulatory requirements in different markets. Establishing clear, science-based regulations will be crucial for industry growth. There are legitimate questions about ownership and bioprospecting that need addressing. When we extract genetic information from extinct plants preserved in public collections, who owns the resulting intellectual property? What obligations do companies have to share benefits with the countries of origin for these plants? Despite these challenges, investments in the sector is booming. Ginkgo Bioworks went public in 2021 at a valuation of $15 billion, showcasing the enormous potential investors see in this field. They're just one of many companies working at the intersection of synthetic biology and manufacturing. Perhaps the most exciting aspect is that we're still in the early days of this revolution. The tools and techniques being developed today will enable applications we can't even yet imagine. Just as the early days of computing gave little hint of smartphones and social media, today's synthetic biology experiments may be laying the groundwork for transformational technologies decades from now. What's clear is that the line between natural and artificial is becoming increasingly blurred. When we used extinct flower genes to produce fragrance molecules in engineered yeast, is the resulting scent natural or synthetic? The traditional categories no longer apply neatly. This technology challenges us to rethink our relationship with nature and history. It gives us a small window into what we've lost through human activity, while simultaneously showcasing how human innovation might help us build a more sustainable future. As we wrap up our journey through the fascinating world of resurrected scents, let's take a moment to appreciate the remarkable convergence of art, science, and manufacturing that made this all possible. What started as a dreamy conversation at Ginkgo Bioworks on whether they could do Jurassic Park for scents has evolved into a groundbreaking project that pushes the boundaries of what's possible with biotechnology. It's given us a sensory connection to plants that disappeared from Earth over a century ago, while simultaneously showcasing the incredible potential of biomanufacturing. Resurrecting the Sublime is more than just a cool science experiment. It's a powerful metaphor for how advanced technology can give us new perspectives on our past, present, and future. It reminds us of what we've lost through human activity while demonstrating how human ingenuity might help build a more sustainable path forward. The fragrant genes of extinct flowers have been brought back to life, giving us a glimpse, or rather, a whiff of biological history. And while we can't fully bring back these lost species, we can use their genetic legacy to inform how we approach conservation and sustainability going forward. For me, one of the most fascinating aspects of this project is how it blurs the boundaries between disciplines. It's not just science or art or manufacturing, it's all of these things simultaneously. It shows how the most innovative solutions often emerge at the intersection of different fields where biologists, artists, engineers, and entrepreneurs can collaborate on problems none could have solved alone. As we look to the future of biomanufacturing, projects like this serve as powerful proof of concepts for what's possible. If we can resurrect the scent of an extinct Hawaiian hibiscus, we also have the possibility of mining that ancient DNA for pharmaceutical products going forward. What else might we be able to achieve with these technologies? The possibilities are endless as they are exciting. In an industry where precision and compliance are paramount, Apprentice's Tempo platform offers a cutting-edge manufacturing execution system solution. It's cloud-based, mobile-first, operator-centric, with the most intuitive UI in the industry, making it perfect for today's dynamic pharmaceutical manufacturing environments and for the production of the world's most complex drug products. Whether you're working on groundbreaking cell and gene therapies, biologics, or small molecules, Tempo provides a fully validated MES system that's both powerful and easy to use. It streamlines your processes, ensures compliance, and adapts to your specific manufacturing needs. So get off of paper batch records and discover how Apprentice's Tempo Manufacturing Cloud can transform your operations by visiting us at apprentice.io. Attention industry professionals, revolutionize your manufacturing process with the Apprentice Tempo Manufacturing Cloud, a manufacturing execution system built to streamline your operations, boost efficiency, and ensure compliance all in one powerful platform. Experience the future of manufacturing with our free 30-day trial. Scan the QR code on your screen now to unlock a world of innovation. Don't just keep pace, set the tempo as an industry leader with Apprentice. That's all for Apprentice.io's Industry Insights. I'm Frank Maggiore. Thanks for listening. Be sure to like and subscribe. Hit the bell icon for notifications when new episodes drop. Leave a comment if you have any suggestions for future topics you would like for us to cover in this space, and we'll see you next week for more Industry Insights.